Before we discuss cardiac output variables and how they are augmented by different variables, let's quickly review the cellular physiology of a myocyte and some intercellular signaling that is critical in understanding how augmentation of these normal processes is possible. Let's talk about gap junctions. We know that gap junctions connect myocardial cells. This is the plasma membrane of one myocyte, and this is the plasma membrane of another myocyte. Travel back to the biochemistry chapter here and visualize the connexons that compose these gap junctions. It is a hexameric protein, so it's composed of 6 elements, and this hexameric protein opens and closes, which allows communication between these myocytes. When you allow communication between cells like this, you can call the network a syncytium. And by syncytium, I just mean that everything is synchronized. So again, when it's open, you allow flow of ions in between the myocytes, and that connects everything to be coordinated. You will see these gap junctions on light microscopy as intercalated discs. These are different from the transverse tubular system, which is known as a T-tubule. Now, T-tubules are essentially just deep invaginations of the sarcolemmal membrane that help transmit the electrical current during a contraction because they increase the overall surface area of the sarcolemma that is able to interact with the extracellular environment. So let's quickly review how myocytes contract. You absolutely must know some important elements behind the physiology of myocyte contraction. Remember that an increase in intracellular calcium causes calcium bind troponin C because it is more available in the intracellular environment, and troponin C loves to bind calcium. That is actually why it was named troponin C. So when we have an increased level of intracellular calcium, it binds troponin C, which causes a conformational change in troponin C. When you change the conformation of troponin C, you also change the position of troponin I. Troponin I stands for troponin inhibitory. When you cause a conformational change in troponin C, you move troponin I from its inhibitory location connected to troponin T. Troponin T stands for tropomyosin because it is bound to the tropomyosin complex. This chain of events allows troponin to couple with tropomyosin, which will move tropomyosin to expose the myosin binding sites on the actin chain. So I need you to understand a key fundamental concept here. The number of interactions between actin and myosin in a cardiomyocyte determines the force of contraction. So actin and myosin together equals contraction. And this is dependent on intracellular calcium and stretch of our sarcomere. More myosin binding sites exposed equals more contractile force. It's like recruiting more people on your team in a game of tug of war. Give your friend some calcium because he loves calcium, and he grabs the rope right behind you to help you contract. There's a slight limitation here also, in that however much you stretch the sarcomere only benefits you up to a certain length. It's just like a rubber band. It depends on how much you stretch it. If you under or overstretch that sarcomere or rubber band, it will not bounce back the most efficient or maximal force that you would want. It can only stretch up to a certain point that you can increase contractility, and this is the concept behind the Frank-Starling mechanism, which we will discuss in the coming slides. So this is an incredibly important image because you need to understand the molecular physiology that is behind the contraction of a cardiomyocyte and how the body augments it in times of increased or decreased demand. If you remember and understand this, you will be golden for the rest of the physiology and pharmacology sections. So as you can see, there's a sarcolemma, which we already mentioned is the membrane of the myocardial cell, and that there's deep invaginations of the sarcolemmal membrane called the T-tubule system. On the T-tubule system, we have L-type calcium channels. We have a sodium-calcium antiporter, which is also located here. We have sodium ATPase. Recall quickly that in phase 2 of our action potential of our cardiomyocyte, there is a calcium influx in this level, and there's also a potassium efflux. These are very important and responsible for our contractions, as well as in the phase 4 depolarization of our SA and AV node, in which the calcium channels set the intrinsic depolarization rate after being activated by the funny depolarization current channel. We will later review the complete electrophysiology, but recall quickly that these L-type calcium channels are what allow the influx of calcium into the cell. So is this what we term the calcium-induced calcium release? No, not quite yet. Remember, the sodium influx from the initial upstroke of phase 0 induced some calcium release, which means that it opened these L-type calcium channels and allowed some calcium to enter the cell. So, am I telling you that this newly released calcium is now going to trigger some more calcium release? And yes, that's exactly what I'm saying. As you can see in your diagram, this RYR is a ryanodine receptor channel. When this newly introduced calcium enters the cell, it will bind and open those ranitidine receptors. And what do you think happens when you change the conformation of a receptor that is sitting on the sarcoplasmic reticulum? Well, not only will it open and allow whatever is stored in the sarcoplasmic reticulum to come out, but particularly it will cause an incredibly large efflux of calcium just to pour out of the sarcoplasmic reticulum. The calcium will go on to bind troponin C, and you get that necessary conformational change in tropomyosin to expose the myosin binding sites on actin, so we get our contraction. Let's go on another step here and examine the other elements of this diagram, which will be crucial in understanding some of the pharmacology later on. Now, what is this PLB and ATP sitting on the sarcoplasmic reticulum membrane? The ATP symbol is representing the sarcoendoplasmic reticulum calcium ATPase, which is SERCA. I hope it's alright with you that we refer to it as SERCA so that I don't pass out every time I try to say the full name. Now, what does SERCA do? Well, it uses ATP to pump calcium back into the sarcoplasmic reticulum, which means that we are increasing our calcium reserves to release next time we open these L-type calcium channels. What about PLB? What is PLB? PLB is phospholamban. This protein is so crucial in understanding how sympathetic stimulation can increase contractility. Phospholamban is a protein that sits directly next to the SERCA pump, as you can see in your diagram. Its job is to inhibit SERCA pumping calcium into the sarcoplasmic reticulum. It inhibits SERCA and says, chill out, that is enough calcium, we're not going to pump any more in there. So basically, PLB, phospholamban, inhibits the ability of SERCA to pump calcium into the SR for future contractions. So if PLB, or phospholamban, is working really hard, it will limit the amount of stored calcium in the sarcoplasmic reticulum. So the next contraction, there will be less calcium stored, and therefore less calcium released. And that means we will have less myosin-actin interactions because there's less calcium available to bind to troponin C, which results in less forceful contractions. So how can you prevent this inhibitory phospholamban from limiting SERCA so that we can get more forceful contractions? What situation would you actually want to do this to have increased contractility in your myocytes by inhibiting phospholamban? How about the sympathetic flight response? Alright guys, take a quick mental image of this diagram. It incorporates beta-adrenergic and muscarinic signaling with the normal physiology of myocyte contraction that we just discussed. Let's catch up to where we were on the last image, when we were examining the protein phospholamban, right here. Remember we said that phospholamban was an inhibitory protein associated with the sarcoendoplasmic reticulum calcium ATPase. It inhibited SERCA's ability to pump calcium into the SR. Now let me add one more important piece of information. Phospholamban cannot inhibit SERCA when it is phosphorylated. As you see on this diagram, with a phosphate on it, it is prevented from inhibiting SERCA, which is the ATP labeled pump here. With this phosphate on, phospholamban is not allowed to inhibit SERCA, and therefore SERCA is pumping calcium for storage in the SR for future contractions. So you may have zoned out on me for a second. So if you did, listen up real quick. This was one of the rare moments in medical school when things click. Do you see the beta receptor sitting on the sarcolemmal membrane? Right here. So the activation of this beta receptor by any beta agonist, in this diagram it's epi or norepinephrine, will activate the G stimulatory subunit of the G protein complex. By doing so, we activate adenylyl cyclase, and we increase intracellular cyclic AMP. Remember that cyclic AMP will bind to the regulatory subunits of protein kinase A and liberate the catalytic subunits of protein kinase A. Basically activates protein kinase A. Now protein kinase A will just go wild, phosphorylating things. There are a couple of things on the slide that you need to know that protein kinase A will phosphorylate. Number one, it phosphorylates the L-type calcium channel, as you can see here. Number two, phosphorylates phospholamban, which is PLB. So it kind of feels like I'm setting you up here, right? Can you tell me which protein kinase A, is probably going to phosphorylate so that we can achieve the overall result that is usually caused by beta-adrenergic stimulation on the heart? Right, nailed it. Phospholamban is the main protein that I'm talking about here. So for completeness also, you must know that protein kinase A will also phosphorylate the L-type calcium channels on the sarcolemma to open them. This means that we open the pore and allow an influx of calcium, and this causes a calcium-induced calcium release. So CICR, calcium-induced calcium release. And by phosphorylating phospholamban, it is no longer able to inhibit SERCA. The SERCA activity increase, and this means increased calcium. Let's summarize quickly. The beta adrenergic stimulation on the heart will activate GS, which eventually will liberate protein kinase A due to the increase in adenylyl cyclase production of cyclic AMP. Protein kinase A will phosphorylate an inhibitory protein that is associated with SERCA. By doing so, SERCA is now not limited from pumping calcium into the SR. SERCA can pump as much as its heart desires, and the calcium will pump towards the intracellular environment of the SR, causing an increase in calcium storage. This is the basis of how all beta adrenergic agonists and antagonists work. They prevent or stimulate this protein phosphorylation cascade. So example, if someone has congestive heart failure and you give them a beta blocker that is specific for the beta-1 receptors, you do not induce the stimulation of this GS. You do not induce adenylyl cyclase to make more cyclic AMP. Protein kinase A is not liberated, and therefore it will not phosphorylate phospholamban, and it will not phosphorylate these L-type calcium channels. You will not have calcium-induced calcium release from those L-type calcium channels, allowing calcium into the myocyte, and you will not have protein kinase A inhibiting phospholamban because there will be no phosphate group added. And this should all make sense, right? Okay, now that we have discussed some important parts of the molecular physiology behind cardiac output, we can begin to individually examine the variables. So stroke volume is the volume ejected from the left ventricle in a single cardiac cycle. You can reason through some things that can change this directly. First, before you can eject anything, you probably need to fill that left ventricle. So the amount of blood that enters the left ventricle, which we call the preload, will directly influence our stroke volume, so preload. Remember, this is related to the stretch of that sarcomere. We discussed the stretching of the sarcomere. The volume of blood that enters the left ventricle will directly determine the level to which sarcomeres in that left ventricular myocardium are stretched. Contractility is the next step to determine how much stroke volume you will have. Remember, contractility is the intrinsic ability of the heart muscle to contract, which is directly determined by the number of myosin-actin interactions, which is an indirect correlation with the level of intracellular calcium. Remember that we can say that because calcium will bind troponin C and it will set off the cascade that will eventually liberate the myosin binding sites on the actin molecule. The last component that determines stroke volume is the force that you have to push blood out of the left ventricle against, and that is termed our afterload. If there is an incredibly high pressure in the aorta, then obviously it will be difficult for the heart to eject blood against that high pressure. Now, what are some things that can cause an increased stroke volume? Anxiety, exercise, and pregnancy. Why? Well, an anxiety attack, people have a very exaggerated increase in sympathetic nervous activity, which is directly going to stimulate these beta adrenergic receptors on the heart. Exercise does the exact same thing. Anxiety and exercise stimulate this beta adrenergic cascade due to the release of catecholamines. And pregnancy is associated with an overall increase in plasma volume, which means that we will increase the preload. So increased plasma volume in pregnancy is due to many hormonal changes, and then this increased plasma volume will directly increase preload and cause an increased stroke volume. So now that we have discussed the things that determine stroke volume, let's examine some other components individually and then discuss ejection fraction. Remember, contractility is dependent on the number of myosin-actin interactions. The number of myosin-actin interactions depends on the ability of calcium to bind troponin C, which will inhibit troponin I from blocking the formation of a troponin-tropomyosin complex that we need to move that tropomyosin away from the myosin binding sites on actin. So basically, the more calcium, the more actin-myosin interactions. The more sites you open up for those two to interact, the more contractile force you will have. So let's go through some things that can increase contractility, which you should automatically think is either an increase in intracellular calcium and the resultant increased myosin-actin interactions, or an increased stretch of that sarcomere. So anything that will activate the sympathetic nervous system will cause the release of catecholamines, which will stimulate this beta adrenergic receptor. If you remember, catecholamines such as epinephrine, norepinephrine increase the activity of the calcium pump in the sarcoplasmic reticulum, here to SERCA. because the inhibitory component of this phosphate, due to the liberation of protein kinase A, that was the result of the beta-adrenergic cascade. Increased calcium allows for more binding to troponin C and moves trypomyosin out of the way so our myosin can bind our actin and we can get that nice, great power stroke. Now, if we go back to this image, a decrease in extracellular sodium decreases the activity of the sodium-calcium exchanger, which is this pump right here. And that means that we will have an increased overall intracellular calcium. So, if you decrease the extracellular sodium, there's less of a gradient, pump sodium in and calcium out. That means that we will accumulate more intracellular calcium, which will allow us to have a more forceful contraction. Again, we see that the level of intracellular calcium is determining the contractility here. Now, digitalis, or digoxin, works in a very similar way in that it will also manipulate the sodium extracellular gradient for this sodium-calcium exchanger here. And it does so by blocking extracellular potassium binding site on the sodium-potassium ATPase. By doing so, you eliminate the ability of this pump to restore the normal high sodium extracellular concentration and high potassium intracellular concentration. So you obliterate these gradients. There's less sodium to flow inward while you pump calcium out. So you basically recreate the situation of hyponatremia because you increase the intracellular sodium because you prevent it from being pumped back out, and therefore you obliterate the gradient for this pump, the sodium-calcium exchanger, to pump calcium out of the cell.